Very good afternoon, respected all dear faculty members, audience, and our honorable guest speaker. So, welcome you all in the day 5 of session 2 of our FDP on innovation in pharmacy education and application of AI enhancing teaching learning practices. For this session, we are having eminent guest with Dr. Subheet Kumar Jain. Before proceeding further, I would like to introduce Sir. Dr. Subheet Kumar Jain is currently working as a professor and former head Department of Pharmaceutical Sciences, founder director, Directorate of Open and Distance Learning, Grievance Redressal Officer, Guru Nanak Dev University, Amritsar. Sir is a leading researcher and academician. He is also the coordinator for Center of Basic and Translational Research in Health Sciences, established under RUSA 2.0 scheme with financial assistance of 10 crore. He is presently member of Academic Council of PTU Jalandhar, MRS PTU Bathinda, and LPU Jalandhar. He is the member of BOS of more than 10 universities of India and known for drafting and suggesting the pathways for implementation of different education policies and guidelines related to education reform. Sir has made India rank 32 and got CSIR SRF fellowship for PhD. Sir has earned PhD in pharmaceutics from Dr. Hari Singh Gour University, Sagar. Sir research area of interest include the use of nanocarrier for sustained and localized delivery of anti-cancer, antifungal, and anti-HIV drugs. Sir work has resulted in more than 150 papers in different national and international journal of repute. Recently, his one of his platform technology patent, sustained drug delivery system, granted in US and another patent granted on previous year. Dr. Jain has excellent H index and very good citation and recognized as top 2% scientist in the list of Stanford University. Sir, welcome at the platform of ISF College of Pharmacy. Welcome, sir.
Thank you very much for such a nice introduction. Thank you, Dr. G.D. Gupta and the entire staff of the ISF to giving me this opportunity to interact with the your esteemed our audience, different researchers, faculty members. So, very good afternoon to all of you.
Good
And
afternoon, sir.
if you permit, then I can start sharing my slides and we can
Definitely,
start.
sir.
Thank you very much. Of course. So I'm just sharing my slides. So I think the slides are visible for all of you.
Yes, sir. Yes, sir.
Huh. I'm just moving the slides. Please check that it is getting moved. Now second slides are visible.
No, sir. No, sir.
No, then I have to keep in that mode only. Just give me time for a minute. So now I think the slides are moving. Please see it.
Yes, sir. Yes, sir. Yes, sir.
Yes. Thank you. So, good afternoon to one and all present here, and thank you for whole ISF family. Special thanks to Dr. G.D. Gupta and Dr. Sant Kumar for inviting me to giving these opportunities. As all of you know that from last 2 years, this AI is changing the whole things. That even we are using the AI in the everywhere. Means in our routine life also, use of the chat GPT, I'm going to be tell you that AI is already being used in the pharmaceutical field, not only from means in last 2 years, 3 years, but it was from the around 30 years back. This AI was even by when this QSAR and other was invented in around 90s and in 2000s. In 2000s, from that particular time, the AI is in the upper means in case of the pharmaceutical field. And here I am going to be take you the some examples where AI is going to be used as a tool very effectively to shorten the drug discovery and development process. Because presently, why the most of the Indian pharmaceutical industries are not going for the new drug molecules, and if you just see the history, that we are having a very few drug molecules, those are contributed. I can say it is only the four molecules that has been contributed by the India in the, and even the last two are in the in last 2 3 years to the
Sir, please share again. I think you are
well,
left out.
म्यूट तो नहीं था पहले सन
Sir, there was no mute, but you left out for some while.
हेलो
Yes, sir.
slides are
Yes, sir.
visible son please
Yes, sir.
somebody
Yes, sir. Yes, sir.
respond
Yes, sir. Yes, sir.
please.
Yes, sir.
Thank you. Sorry. So in this lecture, I am going to be tell you by using the one case study that is for new anti-myopic drugs. Our lab is working from last 10 years. How we have used the this AI technology and how that AI technology are helpful for us for submitting our the data to the US FDA also. So this is the topic subtopics for the my talk, that drug discovery and development process. I'm going to be explain you, and then I'm going to be explain you the use of the computer based techniques in drug discovery process, case studies of new anti-myopic drug, findings, conclusion, and future prospects of the. So I always start the lecture that medicine give life to the patients, but the pharmacists give life to the medicines. And as a pharmacist, we must be feel proud that we are the very important part of this healthcare chain. And in this healthcare chain, there is a significant role are going to be played, especially in case of the drug discovery and the development process. We are the person that is contributing significantly in the different aspects of the drug discovery process, and we are giving the final drug to, as you can say, it's a final weapon we are going to be give. So this is the whole discovery and development process, starting from identifications of the disease and to last, that is the FDA approval. This process start from identification of disease. During the corona, everybody understand this process, that first we are identifying the disease and root cause of that, and what physiological changes this the pharmacists, including the bioinformatics people, including the in silico persons, those who are expert in the other different techniques. They then propose the root cause and pathophysiology of this, that this is affecting the our lung, and in lung it is increasing the viscosity of the mucus, and due to that alveolar and other getting blockage, and due to that our whole means respiratory distress are getting happened. So this was done, and in this process, how the computer based techniques was involved. This was involved that whole bioinformatics techniques was used, whole in silico techniques was also used when we are characterizing the any types of the any molecular pathway of the any drug. And whenever we are even any drugs, when we are going for the new discovery, then the first things that we are thinking, that how, what is the proposed mechanism of action of this drug, and for that we are drawing these schematics. And now these schematics are very much prepared by the use of the different in silico techniques, in which we are doing the all types of the drug receptor bindings, all types of the proposed mechanism, and on the basis of that we are making a one chart and the proposed mechanism, and then we are starting the work. So this is called the identification of the disease. Then immediately we are starting the drug synthesis, or we are going for the already existing drugs we want that we should can use for this purpose. Even in case of the our corona also, initially we have reperfused many drugs. We have tried starting from the chloroquine, and then going to the many antiviral drugs. And presently also this route are in the very much in the demand, that already existing drugs are looking for the new target. Here we this computer based techniques are going to be highly beneficial. Whenever, suppose earlier the screen was presented as in acids, primary use of the screen nowadays is used as you see the market of the screen as in acids versus market of the screen the market is so this the data and the interpretations of the data is going to be very much easy, and this is now a very hot field, even in for future recruitment point of view also, that those who are understanding the bioinformatics, either say additional diploma or by say during the training of their M.Pharm courses, they can be used for they can be trained in this, and every discovery process now, what earlier everything was dependent on SCR. This is the second steps when we are doing after when we are inventing the new drug molecule, and when it is doing all preclinical testings, and when we are establishing the safety and efficacy. Now here also in BPharm, you know that now we are using the XPharm and other software that is simulating the many your in vivo experiment. But there is a one software that is simulating the in vivo experiment, but there are many software are available. Those are predicting the ADME as well as the your toxicity profiling. Both are going to be done by this particular things. So ADME profiling and the toxicity profiling, site of absorption of the drugs, and the extent of the absorptions of the drugs. Now everything are going to be done by the using of the different softwares, and by using this we are predicting that whatsoever the molecule we want to synthesize, what types of the ADME characteristics and what types of the toxicity profilings that particular molecule are having. And if after the in vitro data we are thinking that our molecule are very good in the IC50, any anti-cancer molecule, but we do not know that how it will behave in the body in terms of the ADME. Also we are not knowing that how the 44 different receptor that is coming in our body, it is going to be interact. That's called the receptor paneling. How that molecule are means going to be moved across the body, and what types of the pharmacokinetics characterizations this molecule are going to be have. That everything are now very much possible by using the in silico techniques. And now earlier only the academicians are using these techniques, but nowadays these types of the approach are very common in the the industrial setup also. And even when the people are going for the submission of the some pre-IND doc and other those, in sometimes if they do not have the actual real the lab or the data, in that case they are supplementing that particular point from regulatory point of view from using this means our in silico techniques also. After this, what we have now, we are having a molecule, we have screened the molecule by using the different in silico techniques, and now we have synthesized the molecule also, and we have done the in vitro and the our preclinical testings. And when we have established the IBIBC, in vitro in vivo correlations, and if we are getting the good in vitro in vivo correlation, if the value is coming more than zero 0.9, then we will have the faith that this particular our molecule are going to be behave very nicely when it is going into the in vivo setup. So earlier the success rate from if we are taking the 100 molecule, then only the success rate was less than 1% also. Means even we are taking the 100 molecule, we are not sure that out of the 100, the one molecule that we have taken as considered as a optimized one or the final one, that will behave as expected. So here also in this case our whole in silico techniques are going to be helpful. And earlier, in spite of the checking the 100 molecule, now we are going with the 2-3 optimized one, 2, 3 and 4 optimized one for all detail. And this is saving the lot of cost, and the very importantly the time is this saving. Earlier, whole drug discovery process are generally taking 10 to 12 years period of time, and the total cost involved at coming around 1000 to 1200 crore rupees. And even in nowadays in the present scenario, this cost is touching around 1500 crore rupees. But now this is very much possible in a less cost, means it is going to be done in around 100 to 200 rupees crore rupees, and it can take only 2 to 3 years for getting this finally to the US FDA approval. So this is by using the in silico techniques, we are having a three major advantage. First one is we know our path and we know our destiny, that if we want the ideal molecule, then this should be the path, that we are getting the some information about our proposed mechanism of the actions also. We are getting the information about ADME characterizations also. We are getting the information about the toxicity profiling also. And finally, in place of the checking the 100 molecules, we can able to go only for the few molecules. Those are going to be 2, 3, and 4 molecules. They are going to be good enough for doing this particular thing. So this is the importance of the EI based techniques in this. EI based techniques are not only helpful for here. Nowadays, EI based techniques are helpful for formulation preparations also. That in case of the use of AI in the manufacturing, in the automatic testings of the dosage forms are now very, very popular. You know that what the pharmaceutical industry prepared. The pharmaceutical industry prepared that our all the data integrity should be intact. Means there should be no manual interventions, and now many of the most of the our machines are now fully automatic. You just see the HPLC, you just see the LCMS, you see the LCMS. Now from sample coding to randomizations to the sample picking, analysis, and the interpretation of data are going to be done fully in the automatic mode. You just load the samples, and 24 hours 7 the HPLC and the other they are working. And how they are working, in that case also the AI is getting used. Their microprocessor are the fully means designed and the means programmed in such a manner that it is getting the analysis done and as per the time. Another thing is that nowadays, as per the US FDA requirement, our software should also be the FDA means this should be the SPER compliance. They are giving the, if we are purchasing the UV, they are saying that it is a, you need the software that is a US FDA compliance or the SPER compliance or not. In this case, whatsoever the software they are providing, in that the data manipulations are not possible. So whenever the any audit and other teams are coming, they are going to be check that software, and they will know that when you have done this analysis, what was the sample code, and what was the your result is there. So there was a no manual intervention is there in the data feeding and the preparations of the report. And as you know that India is the manufacturing hub for the whole world, and that even if we want to carry this legacy, then we must be use this AI in our manufacturing and the automation process also. Suppose we are preparing the 50 lakh tablets in a 2 and 3 days period of time. Use of this AI in the manufacturing and the automatic testing of the dosage form, this time is going to be reduced. So this is a one area where the pharmaceutical scientist along with the mechanical and the other software company scientists are required to sit, and we have to tell them what is our requirement, and that requirement are going to be converted in the form of the software. And once it is going to be happened, then probably we are having these fully automatic in the AI based manufacturing. We are having a very much automations in our manufacturing, but in the testing we need means automations. In control of the environmental conditions, we need the better AI techniques, and we also need the better AI techniques for the interpretations of the any types of the future failure in the quality of the our means dosage forms, or maybe in the processes there. In the stability testings and the quality controls, this is very much required, that if we have the some software, in spite of the giving the stability data that we are performing for 4 years, and after the doing the stability for 1 year, if we are having a some interpretations software like the AI based that can interpret it, that by base of the 1 year behaving of this particular molecule, in another 3 years how this molecule are going to be behaved, that is going to be the our future these things are there. So in adverse drug monitorings, we are using the different AI based software. So this is the brief I am giving it, that in none of the aspects of our drug discovery, even collection of the human clinical trials data, even phase 4, when the drug molecule are under the clinical use, and what types of the drugs monitoring and the drug adverse reactions are happening, this all are getting and means managed by the software. Even when we are doing the clinical trials, then nowadays the US FDA and the our good clinical practices are saying that we have to use the software for collection as well as the interpretation of the data. So data integrity cannot be nobody can question that data integrity. And by using these types of the software, you can easily manage the clinical trials in a multi-centric clinical trials. The same software we will use here. Suppose one of the our site is the Amritsar, another site is in the USA, or some other sites are in the Brazil and somewhere. The same software are going to be used, and in this way the whole clinical trial, and nowadays it is a part of our even the protocol also, that which software we are going to be used, and that software is the having a compliance with the good laboratories, good clinical practices or not. So for this purpose also, now the pharmacy background person inputs are very much required to design a software in a such a manner so it can able to capture even the minute techniques for designing of the new anti-myopic drugs, that is the ND10. And especially because our purpose was the formulation development and performing of the preclinical and clinical studies. So I'm not touching that of the target dose profile, and then means optimizations of the formulation, and finally performing and the interpretation of the preclinical data. And especially the histopath is the big issue for all of us, because the histopath we are depending on the somebody else. We have to ask the histopathologist to give the interpretation. But nowadays the many softwares are available. You just put your slides and you will have your preclinical means your that data you will have there in your hand for this purpose. So the preclinical and the clinical, so all three components I'm going to be used. Little bit introduction of the myopia. What is called myopia? Myopia is a vision impairment, near sightness, and due to this why this disorder are happening. Disorder are happening due to the excessive use of these electronic gadget, and the prevalence has been increased. Earlier it was only 14%, now it is touching to 36%. Even in the some of the country, this prevalence rate in the Singapore and in the in Hong Kong and in the Japan and even in the South Korea, this prevalence rate is the 97%. This myopia are happening in the children, and due to this their elongations in the eye is observed, and due to that they have to use the your specs, and they have to put on the some atropine drops and other therapy also. And if this is not going to be controlled, they are having a elongated eye, and that elongated eye in the future, later half of their eye, they can be having a big problem. They may suffer from the many ocular disease, and later on that can also lead to the blindness and other. So this is a very very important disease, and the prevalence of the myopia even in the India also, and in the Asia it is also increasing significantly. So this is the what happening into the myopia. We are having a blurred vision because elongations of the eye is happening there. So one molecule that is called the we called ND10, and chemically it is a 7-methyl xanthine that one of the company has discovered, and they are giving us the task to do the preclinical and the formulations studies for this particular molecule. And this is basically the adenosine receptor blocker, and due to the blocking of the adenosine receptor, it is increasing the collagen's content of the eye, and it is stopping the elongations of the eye. And this mechanism was very proven by the one of the group that is working in the Denmark, that's called the Dr. Trier, and he involved there in this. The things that came to us, that this particular ND10 is having a very short biological half-life. It is having a only 200 minutes, means it is having a 3.5 hours of the biological half-life. And due to this poor biological half-life, means and it need to be administered thrice a day. So you just think that thrice a day tablet in the children, it's very difficult to go, because one tablet they can take in the morning along with the breakfast, then they are going into the school. But the second tablets are when they will come back from the school, then it's going to be evening. So generally they are missing the second dose. Third dose they can take in the later of when they after the dinner also. So due to the thrice a day administrations and due to the poor patient compliances, this particular things are means they ask us to develop the sustained release formulations. They ask us that you just develop a sustained release formulations, and better to control the release of the drug in a such a manner, so the constant plasma concentration of the drugs should be maintained in the body. And due to this, there will be two advantages, major advantage, three advantages. One is the frequency of administration is going to be reduced from thrice a day to twice a day or once a day. Second thing is, it is maintaining the constant plasma concentrations. So due to the maintenance of the constant plasma concentrations, it will having a better control on the disease. This is the inherent advantage of the sustained release dosage form, because whenever we are having a peak and drop type, peak and valley types of the graph, this is indicating. So whenever the drug concentration is falling below the minimum effective concentration, at that time, for that particular window, the drug will not have any effect. And in this case, even we are administering the 3 times a day, but only for 9 hours the drug is remaining above the C minimum effective level. So when we have designed the molecule, then what was the purpose of our designing? Our purpose of designing is this, that we have to control the release of the drug, because as a sustained release dosage form, we only able to control the release of the drug, because it for children and this is for the oral administration. So we selected to develop a simple oral controlled release dosage form, that was the matrix based tablet, and the microsphere both was developed. And the purpose was that anyhow we can control the release of the drug, and that control of the release of the drug was happening only due to when we are controlling the absorption of the drug. Because once the drug, until unless drug was not released, until unless the drug was not solubilized, it will not able to reach into the systemic circulations. And when it will reach into the systemic circulation, only at that time distribution, metabolism, and the excretions process will start. So in this way, we have to what we have done it, we have hypothesized that we are going to be make a floating and mucoadhesive based system, because these are the systems that is retaining and increasing the retention time of the drug in the stomach. And then due to this reason, we will have the sustained release and other effects. So this was the hypothesis. So we will ready to develop a formulation by using the different combinations of the polymers, and those combinations, and our intent was that the formulation should be floating in nature. It should be mucoadhesives as well. It will swell. So as long as is possible, it will going to be remain into the upper GIT tracts for the longer period of time. So this was the hypothesis. Formulation was, but the biggest task for us, because this was the new molecule. So what types of the target release profile we should have? Means whenever we are developing the any sustained release dosage form, the one things that we are determining, that is called the release rate. And that release rate was the very important one. and that release rate, we have to know that what should be the target release, because if I know the target, then my formulation development is going to be easy. If I know that I need to sustain up to the 12 hour, and 1 hour the release should be 12%, 2 hour release should be 24%, on 3 hours the release should be 36%, 4 hour release should be 50%, like this. If I know multiple point release point, and those points should be mathematically, and it should be software driven, I need to put a value of our PK parameters of the IR formulations along with the C minimum and C max value. And if my software will predict it that if you want this types of the release profile, my C minimum was 6.5. So, and I'm having a IR data, and if I fix there that I need the 6.5 micromole concentration, what should be the probable release profile? So this was the output of the software, that if I want to get this types of the future pharmacokinetics profiling, if this is my hypothesis that I need to sustain the release up to the 16-hour period of time, and my C minimum is 6.5, my C maximum is 14, and my therapeutic window is lying between the 6.5 to 14 micromole, then as per the our input of our pharmacokinetics parameters, means Tmax, elimination half-life, absorption rate constant, elimination rate constant, BD, and the dose, I put it here, and I got this types of the release profile. That look, if your formulations are having a such types of the sustained release profile, then you will probably get this types of the in vivo release pattern. This was a big relief, because presently what is going on in conventional way, we are developing the formulations and we are performing the BE study, either in the animals or either in the humans. Most of the company prefer to do in the human, because in the animals, dose escalation and other, they need to perform again. So if the drug safety is not the concern, if the drug safety has been already established in the our by the pharmacokinetic simulations, by using the different experimentations, then they prefer to go for this. But in this way, they are developing the one formulation, they are doing the BE study, observing the results, making the changes, again doing the BE studies, till they are not getting the 80 to 125% of the AUC versus the Cmax profiling. So for this purpose, we have the target release profile for the SR formulations, and this target formulations are supposed to give this types of the profiling is there. So for this software is calculated, and we have developed the prototype formulations, and means only by the 3 formulation my work was over. I have developed the 3 formulations by using this different combinations of the polymers and their amount. And then what I found that this App2 was means that's called similarity factor, and my release profile that was observed, that's for 3 formulation number 23, formulation number 54, 75, you can just take as a number, and that is matching with the target release profile. Point to point correlation is never going to be possible. So always there was one method that is called the similarity factor analysis method that we have performed, and then we found that this similarity factor was found 69.77 in case of these 2 things, and there was one formulation that was not matching. So by this method, initially we are developing the 10 to 20 formulations, but by using this approach and this software, we got only the formulation of development done only in the case of the 3 trials. And this was the profile. This has been plotted as a graph also. You can see that it is almost matching with the target profile. Similarity factor was 69 and 77. So this was a great help for me. That's it. And then we have done the B study of our optimized formulations. We found that whatsoever our interpretations was there, that is exactly matching. Even in the only in the first sort, the B was approved. So you just see that generally for a new molecules in a first attempt, the B is not it's a very rare is getting crossed, because biological systems are highly variable in nature, and due to this highly variable nature, it's not very easy to do in this. But this was a great help. So I told you that how for formulation scientists it's going to be helpful for interpretation of data, for determination of the target release profile, putting the biodistribution pattern, and in case of the formulation development, these AI based techniques are going to be used. Some of the commercially available software, those are doing these all, and the Gastroplus is the leading software for this purpose, and it is very widely used for screening the compound libraries through QSAR and the PBPK data. It is also used for simulations of the molecule, how it will move across the GIT tract, in which particular segment of the GIT tract it is going to be absorbed in the best possible manner, in a which particular segment of the GIT tract it is not going to be have any absorptions. So this will going to be great help when we are designing the formulation. Suppose my drug is not having any absorption in the lobe, means large intestine in the colon part, and suppose I was not knowing this, then what I can do, I probably has designed the simple formulation matrix based. That matrix based formulation are going to be give the sustained release only for 4-5 hour period of time, and followed by there will be no absorption, so the graph will dip, and maybe the my bioavailability will fall very significantly to the level below to the label. So for this purpose, this is a one software that is a very good predicting the first in human and animal dose with vaccine class, IVIV analysis, how the molecules will move across the GIT tract. That is also going to be the judged by this, and it is also going to be give us that what dose we have to test first in human, because from animal dose we are knowing that by multiplying 6.2 and the dividing by the 60 we are getting the human dose, but that human dose are actually safe or not, because first in human is a very critical studies, because first time you are putting anything in the human, that is called the phase 1 study, and if we do not know the risk, then it's going to be big risky if any anaphylactic shocks is there and anything is there. So for this purpose, this GastroPlus is the very good software, and it is having many tools. I'm not in the support of one things that you cannot develop the formulation only by using the computer based or the AI based techniques. We have to use all these as a tools. What is the purpose to use the tools? Tools are helping us to perform any task in a quick and in efficient manner, because sometimes the task take more time, but these tools are helping us to perform that task. So the AI based techniques for us is our tool, not as a final product. You cannot develop even products. If it is possible, you can put in a chat GPT that I need the new drug with this for following 5 characteristics. It follow the Lipinski rule. It should have the dose of 10 mg. It should free from all side effects, and it should have the better A2 receptor interactions and other. That will give you, the software can give you, this is the molecule, you can go ahead, and you can commerce, but it is not happening. If it is happening daily, this software is suggesting the lacks of the molecule, but actual things are when we are doing in the lab, then how this molecule are going to be designed, and how this molecule are going to be behave. That is a very important thing. So for this purpose, we have to use these techniques as a tool, not as a this. Another software is that is called the Membrane Plus. There is a lot of issues that what is the in vivo absorption pattern, how this molecule are going to be absorbed across the GIT tract, how it is going to be the distributed, how the clearance are going to be using. ADME profilings are very important one, and for this purpose, the Membrane Plus softwares are going to be use. This is the entry form software is there, that is called for PKCR. The software also provide the following inputs, that it is used for the percentage of dose absorbed as a function of the time, amount of the solid drug in the GIT tracts, amount of the dissolved drug in the GIT tract as a function of time, and initial it's giving you the some things that related to the ADME profiling of the drugs. DDD plus also the another software, that is it's simulation software for in vitro dissolution experiments. And in dissolution also we are having a lot of issues. When we just see the dissolutions, then the first issue is coming, that which types of the apparatus is most suitable, USP1 type 1, USP type 2, USP type 3, or the 4. 4 has been available, and this is. And what amount of the media we should use? It's a 900 ml, 1000 ml, or whatsoever the amount of the media we should use. What should be the time points? What should be the type of the media? It is a 0.1 NaCl, it is a PBS 6.8, it is a PBS 7.4, that will provide the simulated conditions. So a lot of variability is there, and for this purpose, what should be the RPM? RPM is playing an important role. So it should be on 50 RPM. If I'm using the paddle type, it should be on 75 RPM. It should be on the 100 RPM. So a lot of variability is there. To covering this all variability, what we are doing? We are performing the dissolution in 12 different conditions. And the industry people also need to be performing the 12 conditions. Still, if they are not satisfied from regulatory point of view, then the use of the sinker is also recommended here. So wherever the lot of variability is there, and we need to cover that variability, because the dissolution is the most important parameter that we are considering, and for the in vitro in vivo correlations. So for this purpose, we now the GDT S plus software is there, that used for the simulating the software for your in vitro dissolution experiment of the your any new dosage form, any new drugs, and it is giving you the high quality plots and figure. And on the basis of seeing all those things, you can use as a predictor, and along with the GastroPlus software, you can able to design your the best dissolution profile also. Earlier I told you that we must know the target release profile. Now we have developed the formulations. We must know in which dissolution conditions this particular software are going to be helpful for us. So for this purpose, this software is going to be used for this purpose. So this is also very important from the pharmaceutics point of view. This is another monolith suite is the another software that is used for the your all randomizations and the other things, and then how to do those randomizations, how to done the random, especially in the clinical trials. The randomizations are very important things, and the randomization should be done on the basis of this some scientific parameters. I'm giving you the one examples of that how these randomizations are going to be influence your overall clinical trial results. Suppose I want to perform the 1B studies, and as per the software, my coefficient of variance or values are coming 52. So I have to take the 52 volunteers as in the 1 group. In a B, this is going to be determined. But these 52 human volunteers should be in the 18 to 45 years of the age. That's as per the our standard guidelines for the conducting of the BE study, that this should be in the 18 to 45 years of the age. But this is the guidelines, and they have written that you will women should be avoided if it is it not specific, and if it's some things the very specific is there, you can recruit them as a volunteers. The same are happening in case of the children also. But you just see, when we are recruiting the patients and enrolling the patients from 18 to 45 years of the age, you may have the many variable. First one, the variable of the age in the every age group of the 5 to 7 years. Suppose 18 to 25, how many volunteers are there? 25 to 30, how many are there? 30 to 35, how many are there? For 35 to 40, how many are there? and 40 to 45 is how many are there. So if suppose I will take all 45 years or something, so the software need to mix it them. Huh? The purpose of the software and randomization is this, that in future we have to do in a such a manner so our variability going to be covered. If we not do the randomization, suppose I'm having a out of 50, I'm having a 13% in the age group of 40 to 45, and suppose I keep those all in the same group, probably their metabolic rate in comparison to the volunteers, their metabolic rate that is in the age group of 18 to 25 are going to be very, very different. So for this purpose, randomizations in terms of the your age, in terms of the sex, in terms of their body mass index, in terms of their vegetarian and non-vegetarian diets they are taking, in terms of they are disease free, and from how long they are recruited in these all studies are very, very important. So for this purpose we have to fit this data, and automatically the software will randomize it. That looks this is the A and B, so they will randomize that this is the, you will take the volunteer as say 1 2 3 like this and this thing. So this is the one things AD that is useful in the your profiling of your clinical trial. ADME predictor is also there, and that is also very helpful software for you. And very important thing is toxicity profilings. For whenever we are going for any putting the any data in the for regulatory point of view, 7 toxicity data are important. They are in their checklist. One is acute toxicity data, and that acute toxicity data, what is the maximum tolerated dose in the animals, especially the 2 rodents and 1 in non-rodents. There's a very important one. Followed by what is the effect of the NOEL dose, we call no observed adverse effect level. That is called NOEL. In the acute, we are getting MDT. In a subacute, we are getting the NOEL dose. So for that purpose, we are doing the 28 days toxicity profiling, and we are knowing that suppose in this case it was observed 1000 mg, and our acute toxicity was observed that was more than 2000 mg. So that's called the category 5 molecule safety profiling. So that's considered as a very, very safe molecule in that terms. So for this purpose, acute, subacute, subchronic, chronic, genotoxicity, and mutagenicity, and your reproductive toxicity, these 6 studies are the mandatory one when you are filing any applications, even for phase 1 clinical trial, even for any of the regulatory agencies. They need these studies. So here is, these softwares are very, very helpful for us. That's for predicting it, and we have used this software, and then we have determined this, and the tool name is there because we have done these all 6, but they are asking for carcinogenicity data also, and HERG inhibition data also, how it will affect our heart rate, and they are asking for detailed hepatotoxicity, they are asking for nephrotoxicity, they are asking for cardiac toxicity, they are asking for blood-brain permeability, because these are the 5 major organ of our body, and they are very concerned, regulatory are very concerned about the hepato, nephro, cardio, liver, and the brain and toxicity, and the reproductive toxicity. We were not having a data, so we have done this by using the ADME tool, and this carcinogenicity data and other we have included as a supporting document there, with the undertaking that in the future we will do it as a, when we will do the real time and the phase 3 clinical trial, simultaneously we are going to be evaluated them. And this types of the this was the great help, and they have accepted it, and now it is going for the phase 2 clinical trial. These are the some things, some interpretations that carcinogenicity and the heart toxicity is called HER inhibitions. That data is there. Another issue is coming, that is a very, very important, that what is the drug-drug interactions. When any drug molecules that ready for launch, and along with the toxicity profiling, and along with its efficacy profiling, along with its dosage form detail, we have to give the one more information, that is called the DDI interactions, drug-drug interactions, from which particular type of this particular molecule are having, probably is having the interactions, and this particular and that drug-drug interactions is how it is going to be so what should be the recommendation that it will recommended to give with the means corticosteroids or the any of the other class. Even you just see like the warfarin is not recommended to give with the sodium valproate. There's a many non-drug interactions. Tetracycline is not recommended to give with the any of the calcium-based formulation. This has been written in their leaflets. So for this purpose, how this has been written, they are asking to perform this. So if we are performing naturally at the beginning, we do not have this type of the data. So at that time, this particular data can be predicted by the this particular AD software, and assay information we can do it, and by short with the very short number of the drugs, we can able to do this our particular molecule. When we have screened this through software, we found that this molecule are going to be having an interaction probably with the around this 10 drug out of the 500 I screened. We have screened this molecule against the all existing drug. That database was having a all approved clinically used drugs that database was having. So in that case, we have screened all across that, and we found that only across this 10 drugs it is having a probably the interaction. So now we are performing these things. Another thing is histopathology is very, very important, and you know that ultimately even in case of any toxicity and other along with the quantitative parameters, the histopath parameters are very important. For us pharmacists, we are helpless. We are dependent on the some medical people to, and especially not only the normal medical people, those who are pathologists, they have to evaluate this and they have to give interpretations, and such expertise are available with the very few people. Even their interpretations are going to be very different also, and due to this interpretations, we are not able to take the right decision. Some are saying it's safe, some are saying not. So for this purpose, this is the another tool that NAFL this system be in which you just put your slides photographs, and we have put it we have because we have done this molecule, we have evaluated for the treatment of the NAS also, hepatic disorders, and then this AI tool has giving us the interpretations as the pathologists are giving us, and that tool was very helpful. Sometimes they do not have time, so we are using this particular data. So this is also very, very helpful tool for all of us. These are some other softwares, those are also going to be used. So in the last, just I want to tell you that use of AI based techniques are from beginning to last. I told you, starting from the when we are starting the invention of the drugs, then doing the QSAR analysis, shorting the drug molecules, predicting their ADME profiling, predicting their drug receptor interactions, predicting their their future ADME profilings, helping into the formulation development, predicting their stability profiling, helping to base use of the AI-based techniques for development of the dosage form, and in manufacturing at a very, very fast rate, conducting the clinical trial, randomizations of human volunteers, coding of the placebo and the active drugs, capturing the data at a real time basis, analyzing the integrity of the data, and then finally doing the interpretations and the statistical treatment of the data. and also getting the future directions. Everywhere nowadays, this AI based techniques are going to be helpful, and this is probably if we capture these all things, it's going to be our future. And nowadays, even everywhere, even in the drug interactions, drug excipient interactions, even in the type of the formulations, everywhere the AI based tools are there. So we have to use only as a tool for this purpose, and this is basically the tool. That's not the full package. Once we consider it's a whole package, probably then in real time we will face a problem. So as a scientist, as a formulation scientist, we must understand that this is a tool, and the tool are going to be help us to do perform the task in a very quick and efficient manner in a very short period of time. So this is my message to all of you, that these are the very great helpful tool. And as a pharmacist, future in our future, probably we will also having a some job in the software company also, that just to guide them that what is the requirement of the pharmacy, what types of the software need to be do. So this is also giving the another era to the pharmaceutical scientist. If they understand this, then probably it's going to be helpful. So thank you very much for all of you for patience listening, and now this is open for if any doubt is there, and then I'm happy to answer those things. Thank you, Gupta ji, for inviting me, and giving me this opportunities. Also Dr. Manish, and the whole team of the our ISF, also the all the participants. Thank you very much.
Thank you, sir. I would like to invite our director, sir, for comment on the lecture. Sir, please.
So, good afternoon, everyone.
Good
So,
afternoon, sir.
first of all, I am thankful to today's very dynamic speaker, Dr. Subheet Jain, and he has given the very important technical information and the knowledge related to the artificial intelligence from application tools and each and everything from drug molecule to the dosage form. So obviously nowadays Pharmacy Council of India recently they have announced the new syllabus as per the NEP 2020, and they also highly focused on the artificial intelligence. So no doubt these types of the lecture and these types of the things, it will be the very helpful to the students and teacher in the academia research and development of the new dosage form, and also application in the drug molecules drug discovery, as well as AI is the application of the even is the more important in the dosage form design, as well as the administration like part. This is the also helpful. So I hope that we know that the pharmacy peoples, those are the very senior and they are the involved in the research. They also can be nowadays many peoples they are the collaborating with the IITians, some of the electronic electrical persons, so that this types of the electronic system it will be the convert in the form of the application. So obviously the purpose behind of this FDP to connect the peoples to provide the knowledge related to the artificial intelligence, especially in the application in the pharmacology, pharmaceutical chemistry, pharmaceutics, as well as the pharmacognosy. So during these whole of the FDP, this is today is the fifth day. We have invited the people from different field, from the industry, from the chemistry, from the ecology, from the sotex, and each and every person they have the given their the brief knowledge related to the application in their zone, especially in the pharmaceutical sciences. Nowadays, number of the area it can be also available to work in the pharmacy practice, and peoples are the doing the lot of work. So you have given the very specific knowledge, technical format, as well as you have opened the scope for the many tools you can utilize during the research system. And if you have the more and more, then you can develop your own tool for the application of the drug delivery system and the dosage form design. So thank you very much, and thanks to the all the organizing committee member to provide this opportunity to interact such types of the speaker, and thankful to the all audience, those who are the connect direct on the YouTube our Google Meet as well as the YouTube channel. So Dr. Subheet ji, all lectures they are the available on the YouTube channel. So more and more people they will take the benefit related to the lecture, because I know that each and every students, faculty member on institute, it is very difficult to organize such types of the FDP. So these types of the potential lecture always helpful to the people. So once again, thank you very much, and for accepting our invitation and provide a very purified and justified knowledge and application of the artificial intelligence. So thanks to all. Thank you.
Thank you, Gupta ji. And it's your very good initiative that you are keeping these all lectures on the public domain. So in the future also, if anybody want to take the advantages of this, so this is a real serving of the pharmacy, and you are doing from last 30 years, and hopefully you will continue for many more. Thank you very much, sir. Thank you,
Thank you, sir.
ISF
Thank you,
for
sir.
inviting. Thank you.
Thank you, sir. Thank you, sir.
Thank you, sir.
So I'm leaving, sir. Thank you very much. Yeah. Take your lunch.
Thank you, sir. Thank you.
Thank you, sir. Thank you.